One so slightly different topic, but the way it is relevant to genomics is that the methods that we use for viral genome analysis, we have employed both on whole genomes for human genome analysis as well as on several bacterial genomes. So the methodology, some of them are directly transferable. So I was at CMU Qatar as a visiting faculty. Now I'm back at University of Pittsburgh, where I'm an associate professor. And so the topic is analysis of inverted repeats in viral genomes at large scale. Um, sorry. Okay. Palindromes. So you all know what palindromes are. They read the same forward and backwards, or at the midpoint, they're the same front and back, like race car or able was I or I saw Elba. Do you know that this is there's an entire story written in Telugu, which where every poem is palindromic. So Telugu is a morphological language, so it may not be very super clear, but you can see that it is the same, right? So these interesting patterns, these patterns are interesting to find for various reasons. And in genomes, they are interesting because they have a functional relevance. So the DNA palindromes are similar to regular palindromes, except that the reverse one is a complement of the forward one. So you have a G and a C, a T and a A, and so on. And we have done previously DNA palindrome analysis on the 1,000 Genomes Project data and release them for users who study the biology of these things. And here I'm presenting similar work on viral genomes. Inverted DNA inverted repeats are similar to palindromes, except that you allow for a gap sequence between them. It can be zero length, that is, there's no gap sequence, or it can be as long as even 10,000 base pairs. So they are believed to be relevant in viral genome, the way it inserts itself into the host genome, DNA, and so on. And it's believed that they are abundant, but how abundant are they? So that's what we wanted to find out. While there are a few examples of viral inverted repeats, like adeno-associated virus has a big repeat, which I'm going to show you. Zika virus also is supposed to employ inverted repeats in developing diversity and adaptability and through evolution. Adeno-associated virus has a humongously big inverted repeat on both terminals. So this one, as you can see, is a complement of this one. It forms a T junction like this, where B and B prime are complement, C and C prime are complement, and you have an identical pattern on the other end of the virus. So are there other viruses who have that type of inverted repeats? So that's the question. We wanted our software to be scalable and flexible in terms of how big the half lengths are and how much of a gap sequence is allowed between them. And so our approach is actually, so many people came up with, not many people, there are only like 4 or 5 big software tools. But the point is, people come up with various methods. What we used is an interesting observation that if you have a sequence like this, and if you look at the complement sequence, that is a complementary DNA strand, it'll be an identical sequence. So whatever you have here, you'll have it identically on the other side. So instead of looking for complementarity, we can just read the 5' to 3', read again 3' to 5', and then look for identical matches. And we used this biological language modeling toolkit that I developed long ago to build what is called augmented suffix array. I don't know how much time I have. I thought it's a long time since people have talked about suffix arrays, and I wanted to explain them a little bit. I'll just briefly explain the concept. So you have, if you have a whole genome sequence, you are considering suffixes from every position all the way to the end. And if you consider, so the suffix at position 13 is GGC going all the way to the end. And then you arrange them lexicographically. This can be done with linear time, linear space requirements. And then you'll find what is called a longest common prefix array, which is how many letters are common from one suffix to its previous suffix in the lexicographic ordering. So that is very useful. So I developed this. So the first speaker said that, you can look at the aspect ratio of the slide. As you can tell, I'm very old too. But not only that, the software I developed when I was a student, the students who worked with me in analyzing them are born after the toolkit was developed. Okay, so I worked with a few undergrad students, and those undergrad students were born after the toolkit was developed. So how we find this is basically, as I said, read the forward sequence, read the reverse sequence, and then you go find exact repeats in that using the suffix array toolkit. So the results, basically, we compared our method. The orange one is our method, and these are a couple of other methods that have been there. It's not a very widely studied problem. There have been like something in 2000, something in 2008, 2010, but there are not that many tools. So the few tools that are out there, we compared it on 5 viruses, found that BLMT is performing better. So these green ones are common to all the tools, and the purple ones are unique, and the blue one, which is practically non-existent because there's only one extra thing that they found, are found by other tools. So this is just showing them on the same scale as a percentage. And then we applied that to 14,000 viral genomes. We had several interesting things come out of it. Some viruses like monkeypox virus and so on, they are known to have inverted repeats. So we found them, and then we found other viruses out of the 14,000 that also have a large distribution of viruses. Large distribution of inverted repeats. Some of them are very, very big. So we also analyzed in terms of what the host virus is and so on, and found interesting patterns that, this is how the distribution of the inverted repeats is. And I thought I had one more slide. So the other slide, the other result that I have on the poster is that there are very, very big, like thousands of base pairs of inverted repeats in the viruses. And clearly they play a role in some of the viruses. We already know that they're playing a role in how the virus inserts itself or controls the gene expression and so on. Controls the viral packaging and so on. And the question is, what other viruses can we study using the tools that we have? As I was going to say here, that this tool, the software suite that I developed is so ancient that I used to call it Patternix Revelio. I don't know how many of you read the books of Harry Potter, not the movies, the younger ones. So the people who read the thing, that there's there is a spell called Specialis Revelio. My goal was the toolkit should be like that. We don't know what patterns we are looking for, but we want to run it on whole genome sequences and find all kinds of patterns. And if you're interested in talking about it, please do contact me. All of these are undergrad students. I also want to point out that Madhura is from India. Preet is also from India, from a different institute. George is from Philippines. And Eric Jingxiang Gao. Eric is from Qatar. So they all wrote to me saying, hey, I want to work with you. And they're all working independently as undergrad project students to analyze the results that we produced with BLMT. Professor Matthew Strawman is the one who asked me saying, hey, Madhavi, I believe you know palindrome finding, and I know adenovirus. Do you want to find out whether there are other viruses that have similar inverted repeats? So basically, I pursued this problem because he asked me that question. That's all.